sí, sí, hola, sí, hola, sí. Sí, sí, hola, hola, sí, hola, hola, sí, sí, hola. Sí, hola, 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 sí, sí, hola.
Welcome to the University.
The people with the camera here recording, they are expecting that okay, not to move too many. And there are two water bottles here.
Okay.
One is for her. And
One over here.
yes. Okay. Thank you very much.
Permiso.
me dices qué hacer.
¿Lo queráis? ¿Empiezo? Good afternoon. Please, may I have your attention? Good afternoon, everyone. It is a great honor to welcome you to the first World Congress of Faculties of Pharmacy here in Salamanca. The Congress is included in the celebrations of the 8th centenary of the University of Salamanca, which is introduced in this video.
Welcome to the University of Salamanca. Come in and discover a history of 8 centuries, its rich artistic heritage, its long humanistic and scientific tradition, and its ever-present universal vocation. Come into its modern and functional campuses. Share an interest with more than 30,000 students. Walk through its 26 teaching centers, 16 faculties, 3 advanced technical schools, 6 colleges, and its School of Aeronautics. Gain access to knowledge through its wide-ranged and abundant course offerings. The University of Salamanca captures talent and gives back to society expertise, innovation, and culture. Support its research groups. They are like engines of development. Their activities reverberate in our society and economy. They promote employment and the entrepreneurial spirit. Get carried away by the flow of a campus of international excellence, by its momentum in teaching and dissemination of the Spanish language and biosciences, and by the creativity, dynamics, and critical commitment. We are all counting on creating an effective, efficient, and responsible university. Welcome to the university.
Thank you. On first place, to present this pharmacy congress, let me give the word to Dr. Antonio Muro, President of the Organizing Committee and Dean of Salamanca's Faculty of Pharmacy. Please.
Thank you, Raquel. Good afternoon, political and academic representatives, students, attendants who join us from all over the world. Welcome to the city of Salamanca. Welcome to the University of Salamanca, place where we convene today to celebrate the IPAP Congress, Innovation in Pharmacy, Advances and Perspectives. I'll be brief, and as per usual, my words could be summarized in 3 sample ideas. Their common denominator is one, and it revolves around one word: gratitude. First and foremost, I'd like to say thank you to the past. In January of 1218, the King Alfonso IX of Leon founded the General Study, establishing disciplines such as law, medicine, or music. Subsequently, Pope Alexander IV granted worldwide validity to studies course in the University of Salamanca. Women such as Beatriz Galindo, probably the first female university student in the world, or Lucía de Medrano, who imparted classes in the University of Salamanca, are 2 extraordinary examples of the vital role that women have had in this university. Likewise, great professors have also left their mark. As an example, I'd like to mention Antonio de Nebrija, who published his Castilian grammar in 1492, first of any Romance language in Europe, or Francisco de Vitoria, who in his class explained the rates of the indigenous peoples in the American continent. The Faculty of Pharmacy was created well advanced the 20th century. I'd like in this moment to thank the deans and their respective deans for their remarkable work in making this faculty, one of the most currently relevant in our country, hoping that in the coming years it will achieve a broader international presence. Secondly, it's now time to show our gratitude to the present. After many years of hard work, we gather here today, inaugurating this Congress, in which many of us have invested our energy with hopeful anticipation. We couldn't have had it any other way, as we celebrate the 800th anniversary of this university. Consequently, my first wave of gratitude goes to all of those who comprise the organizing committee, deans, dean and coordinators, administrative staff, students, the technical secretariat of the University General Foundation, and along, etc. Week after week, we have gathered to design and formulate this complex Congress with the ultimate objective of debating about the advances and future perspectives in pharmacy, those relating to education, research, and pharmaceutical projection. I also want to take this opportunity to thank the organizers of the 40 workshops with the participation of 80 speakers who have joined us from 22 countries. I personally want to show my appreciation to the plenary speakers. Phillip, thank you. Mariano, thank you very much. Manuel Elkin and Beatriz. It's a true honor to have you here with us. With very warm and special thanks, I am delighted to highlight that the 1993 Medicine and Physiology Nobel Prize winner will deliver our opening conference. Thank you again, Phillip, for your kindness and generosity. This event couldn't have happened without the support of the 32 sponsors who have contributed their efforts, resources, and collaboration to this Congress. Permítame que cambie a mi lengua materna para agradecer a la Conferencia Iberoamericana de Facultades de Farmacia, COIFA, por haber elegido a Salamanca para la celebración de la reunión anual de la Comisión Permanente, a la Conferencia de Decanos por haber incluido en este evento el Congreso de Educación Superior de Ciencias Farmacéuticas EduFarm, y a las Academias de Farmacia Iberoamericanas por haber tenido la gentileza de celebrar sus actividades dentro de este marco. Expreso mi agradecimiento muy especial a las facultades de farmacia que han elegido a sus estudiantes, los mejores, para presentar los resultados de los trabajos fin de grado. I'd like to close this section by thanking all the attendants who join us today from all over the world. I hope that your expectations are met, and above all, that you enjoy your stay in this beautiful city and our outstanding university. And we have reached the point to discuss the future. I'd be enormously grateful if this gathering would become a starting point for international collaboration with the foremost goal of improving health and reducing suffering and disease. Termino ya, no sin antes citar 3 de las frases recogidas en el juramento del farmacéutico al finalizar sus estudios de grado. Trataré a todos los que sirvo con igualdad, justicia y respeto, independientemente de su sexo, raza, etnia, religión, cultura o creencias políticas. Apoyaré el avance del conocimiento científico y de la práctica farmacéutica y aprovecharé las oportunidades que surjan en mi entorno para colaborar con todos los profesionales de la salud. Thank you very much. Obrigado. Gracias. Welcome and enjoy the Congress. Thank you, Antonio, for your words and for making possible this Congress. Intervenes at this moment Dr. Carlos Treceño, President of the Council of Pharmaceutical Colleges of Castilla y León.
Students, deans and teachers of the University of Salamanca and other universities, academicians of the Academy of Pharmacy of Castilla y León, National Academies of Latin America, representatives of scientific societies, presidents, members of the governing boards of other chambers, fellow pharmacists, authorities, Good afternoon. I wish to thank you, the Dean of Pharmacy of the University of Salamanca, Antonio Muro Alvarez, President of this Congress, for his invitation to the Regional Council to be present at this first World Congress of Pharmacy, and my congratulations for the excellence of the content of this Congress. And of course, I would like to highlight how delighted I am today from being able to listen to Professor Phillip Allen's harp below. It is an honor for me to share a table with those who accompany me today and to take part of this event, representing all the pharmacists of Castilla y León, more than 5,000 members who work in their different professional fields, the majority, almost 73%, in community pharmacy. This represents a unique healthcare meeting in which university students and professions come together, coinciding with the commemoration of 800 years of history of the University of Salamanca, the oldest in Spain. In an environment such as Salamanca, a reference point for culture, a student destination par excellence, and home to the only faculty of pharmacy in Castilla y León, in fact, a large part of the members who work in our community, among whom I am, have been trained in these classrooms. With the slogan, Innovation in Pharmacy, Progress and Prospects, we set the starting point to a Congress with an excellent scientific and professional content, full of actuality, of professional practice, and looking toward the evolution and the future, because pharmacists are part of a living, dynamic profession which advances. For some years now, our environment has been undergoing important socio-demographic changes that affect the society in which we live and our health system. The aging of the population, epidemiological diversification, educational change, and new technologies are some of the changes that have a notable influence on the role of health professionals in the 21st century. In this context, pharmacists and pharmacy, as an integral part of the health system, need to adapt to change and move forward with the medicines as a tool and the patient as an objective to meet the needs of both patients and society in general. For this reason, graduate training of those who will be professionals of the future requires not only quality university training, such as the one provided in this Faculty of Pharmacy, but also continuous training and constant connection with the profession. So, I hope to have the opportunity to see you again in the Second World Congress of Faculty of Pharmacy. Let us continue to innovate and give society and health the best of ourselves, our knowledge of medicine and health. Thank you very much.
Thank you, Carlos, for your kind words. It is now the turn of Doctora Beatriz de Pascual, President of the Spanish Dean's Conference of Pharmacy Faculties.
Good afternoon. As the President of the Organizing Committee has previously mentioned, we celebrate also here the 6th International Congress of Higher Education in Pharmaceutical Sciences, EduSpharm. It begins here at the University of Salamanca, but the first edition was held in Barcelona in 2006, and then it was organized at the University of Santiago de Compostela, together with the celebration of the 150th anniversary of its foundation. Then came Granada, La Laguna, Sevilla, and Navarra. EduSpharm is a wonderful forum for professors and students of pharmacy to share our experiences and teaching innovation projects. There are many changes in recent years in the field of teaching pharmaceutical sciences. Implementation and accreditations of degrees in pharmacy, acquisition and assessment of competencies conferred by the degree, collaboration with professionals in the field of pharmacy and hospital pharmacy, for the assurance of quality supervised practices, introduction of our undergraduate and graduate students to scientific research, internationalization of our degrees, and many other topics. Sharing ideas and experiences between teachers and students enriches us all and helps us to harmonize procedures and strategies, so that without prejudice to maintaining the style of each university, we can work together in a global environment for the good of the future professionals of pharmacy. Throughout these last 5 editions of EduSpharm, the organization has improved and nowadays attracts an increasing number of students, researchers, professors, and pharmacy professionals from all fields. We now initiate Salamanca 2018, where we celebrate the 800th anniversary of this university. The president of the organizing committee, Professor Antonio Muro, Dean of the School of Pharmacy of this university, has been daring and brave enough to remarkably increase the relevance of this event. He has even chosen the perfect date for the opening session, the 25th of September, the International Day of Pharmacists. He has included a significant number of activities involving students, lecturers, researchers, professionals, and also very interesting speakers from all fields. To reach this goal, he's got the involvement of a large number of members of the organizing committee who have made a wonderful and tremendous effort. I would like to thank them all for their dedication, and of course, I want to thank Professor Antonio Muro for coordinating such an impressive version of EduSpharm. Dear Deans of the Schools of Pharmacy of Spain, the overwhelming program that starts in a few minutes with the opening session with Professor Phillip Allen Sharp constitutes a challenge for the organizer of EduSpharm 2020. However, I am confident that you are all completely and enthusiastically involved in our common goal of improving the education of the future professionals of pharmacy, and therefore this EPUB 18 will constitute a springboard for discussion and further development for the benefit of the students in our 23 schools of pharmacy in Spain. I hope you enjoy the program. Thank you very much.
Thank you very much, Beatriz. It is now, next, we will listen to Dr. Ivan Torres, President of the Ibero-American Conference of Pharmacy Faculties.
Good afternoon, authorities, ladies and gentlemen, participants of this magnificent event. It's a great honor for me to be here. Let me thank the opportunity to address you on behalf of the Ibero-American Conference of Faculties of Pharmacy, institution with 25 years at the service of pharmaceutical education, and that brings together more than 50 institutions with representatives in 20 countries, 18 from America, Spain, and Portugal, and reaffirm our commitment to continue working together, since I believe it's time to join efforts with the major purpose of contributing to the improvement of pharmaceutical education in Ibero-America, and we have a result now. It's the approval of the proposal of minimum curricular contents in a framework of pharmaceutical harmonization here in Salamanca. I'm sure that together we can achieve the objectives we have set. And now, like Antonio, let me change. para expresar nuestra gratitud al rector magnífico de la Universidad de Salamanca y al gran equipo liderado por Antonio Muro en la organización de los eventos. Un gran evento que nos congrega y nos permite mirar hacia el futuro para forjar juntos lo que queremos para la farmacia. Más aún en un día tan especial como hoy, en que celebramos el Día Internacional de la Farmacia. Thank you very much.
Thank you, Ivan. Finally, to inaugurate the Congress, I give the word to Dr. Ricardo Rivero, Magnificent Rector of the University of Salamanca.
Thank you and good afternoon, authorities, speakers, and participants in this International Congress. Welcome to the University of Salamanca, where we are celebrating our 8th centenary this year. The organization of large international congresses such as this one, Innovation in Pharmacy, Advances and Perspectives, demonstrates our firm vocation in favor of research. I want to thank the Faculty of Pharmacy, in the person of his dean, Dr. Muro, and the participating departments, institutes, and research groups for all their effort to make this meeting a success. The progress of societies and the quality of life of human beings is closely linked to scientific advances. Innovation is the result of processes for improving cultural, scientific, and technical solutions to people's problems and needs. Historically, the pharmaceutical sciences have made great contributions towards these objectives. The University of Salamanca has excellent researchers in the area of pharmacy. The Faculty of Pharmacy is among the best rated in Spain. Our students receive a first-rate education. We also try to inculcate the values of the University of Salamanca: effort, progress through knowledge, and scientific curiosity. Classical works, such as Dioscorides, can be found in our library. Our researchers continue to make contributions at the frontiers of knowledge in cardiac and brain therapies, pharmacogenetics, nanotechnology, big data, and nutrition. Thanks once again to the organizers, and I wish you every success in this meeting. Muchas gracias a la organización, mucho éxito y viva la farmacia en este su día. Bienvenidos.
It is now my pleasure to welcome Professor Philip Allen Sharp, who will be introduced by Dr. Mariano García Blanco, Professor and Chair at the University of Texas Medical Branch. Please, Mariano.
Buenas tardes. Now I switch to English. So it is a personal honor and privilege to be here. I want to start by thanking Antonio. And tomorrow, I'll tell you what it means to me to be speaking in Salamanca. But today, it's about Phil. So I could give you the Wikipedia version of his very long and distinguished bio, or the Britannica, if you believe in the encyclopedia. But I'm going to actually summarize it so we can get to the really important thing. So I'm going to summarize it from the point of view of a university. So Phil has been at MIT for 44 years, correct? Where he is the highest level professor you can be, the institute professor. And his career there can be summarized in 3 words. I'm going to start with what I think is the most important one: science. Second one is mentoring. And third one is service. In science, I think we will see a lot of very interesting things. I think we could talk about prizes, national academies, the US National Medal of Science, Lasker Award, the Nobel Prize in Medicine or Physiology. For some scientists, and I'm venturing a guess here, for some scientists, that will be the highlight of your career. For Phil, it was a joyous and important step, but he hasn't stopped one moment since then in continuing up. But the other important point I want to make about science is that he has not only done fundamental science, he has also been remarkable in translating those results and making it impact society. Those are very important. I think for a modern university, you need both. Mentoring, very little can match his record. As some of you know, he has a graduate student who was already a Nobel Prize winner. So his mentoring prowess is impressive. Those of us who have been his mentees are lucky to have had that experience. And then the third is service. He has served MIT, an institution that he truly loves, extremely well. Not only has he been a professor for many, many years, he was head of department, he was head of the institute, head of the cancer center. This is what every university would love to have, is somebody with that type of dedication. So as you listen to his science, think about all the other amazing things he has done. So it is a great personal or professional honor to introduce Phil, and we have to be informal, to give this talk. Welcome, Phil.
Thank you, Mariano. Little did I know when I had you in my lab that you would introduce me in Salamanca. It is an honor to be invited to address the Congress of Pharmacy. It's an honor to be here on the 800th anniversary of this university. And it's an honor to be able to address the students and the faculty of the university. I work at MIT, it's 150 years old. And to stand here and think about 800 years of educating students and advancing knowledge is just awestruck for me. But what I want to talk about today is a particular view of transforming pharmacy through scientific advances. I will primarily talk about how fundamental advances in science has, over time, translated and changed our understanding of science, but as well, and equally important, changed the lives of patients who pharmacy is part of that translational process between science and patients. And I will primarily talk about the translational side, and I want to begin with a little history. Molecular biology was a fundamental advance in science, as we will mention in a moment, happened in the mid-50s. And if you think about it, after the development of crude vaccines and small molecules as drugs, it wasn't until the development of molecular biology that we took pharmacy and we took our science to a new level in benefiting patients. And I will talk about that translational process between molecular biology and recombinant DNA. And I will note that I have a conflict of interest with a company called Biogen, which I will talk about and mention in these comments. I will also follow that with the discussion of how a new type of pharmaceutical agent is being developed to treat patients. And this comes out of the science of RNA interference and antisense technology. And the leading company in the RNA interference field is a company called Alnylam, and I'm associated as a founder of that. And then finally, I will turn to what I think is the most new and exciting development in molecular and cell biology and spend a few moments trying to convince you that there's still a great deal to be learned in this field and it will, in time, transform how we deal with healthcare in the world. Let's think back. In 1953, Watson and Crick, with data from Wilkins and Rosalind Franklin, discovered the structure of DNA. And that gave us, in the first time in the history of man, a view of how information was transferred between generations in this beautiful molecule of double-strand DNA. And that is the origin of molecular biology. It really was a biology of information transfer between generations and a focus on this wonderful molecule of DNA that had that sequence and information in part of a linear sequence. I was 9 years old when this discovery happened, so within the span of my life, we're talking about this science and what it's meant. It took us another 8 years to understand how the information in DNA was first transmitted into the cell in the structure of an RNA, which you see is that dark line below the DNA, and then how that was translated to make these functional proteins that carry out the metabolism of the cell and all the other processes in the cell. And this is from the work of the great French scientist Jacobin Monod, and advanced molecular biology to the point of understanding how genes were expressed and how they were regulated. But most of this science had been done and was done until the mid-70s, focusing on genes and gene activities in bacteria. And as Monod said, what was true of elephant is true of E. coli and vice versa, a bacteria. Well, he overlooked a few simple facts. In the mid-70s, it was discovered that the structure of genes in our cells was actually very peculiar. It had this discontinuous split gene structure. And that's illustrated here in this slide, where you see the central line is DNA. You see those boxes, and I'll call those exons. And those exons have the information that's encoded in the gene. Between those exons, you see DNA sequences that are dashed lines. Those are called introns, and those do not contain information for the proteins that are made from this gene. So what has to happen, and happens at the stage of RNA, is that the black exons are spliced together and those nonsense sequences within the intron are removed. But you notice when you look at this gene that there are several patterns, pattern 1 and pattern 2, that give different combinations of those boxes. Those are called alternative splicing, meaning different sets of gene sequences are used to make different proteins from one gene. Though we have only 23,000 genes, we express, due to this alternative selection of exons, a vast number of different proteins from each of those genes. And it may teach you a little humility to know that many of the insects flying around out there have a lot more genes than you do. We just use them in a more complex way than they do. In the mid-70s, with this discovery and the discoveries of recombinant DNA and DNA sequencing and chemical synthesis of DNA, we gained the ability to take genes from one species and introduce them into another species. We obtained the ability to do synthetic biology for the first time. And this led to the possibility of taking genes that are critical for different disease patterns and different resistance to disease, and move those genes into bacteria and make large amounts of protein from the rapidly growing and high biosynthetic capacity of bacterial synthesis. And this science had developed exclusively in laboratories around the world on research almost exclusively on trying to understand the nature and the function of genes. And a number of us understood that for this science to actually be translated to make a difference, one had to move it into the private sector in which there were resources that would allow teams to be developed that not only understood genetic engineering and recombinant DNA, but understood patient needs, understood how to produce these proteins in large amounts, and understood how to do clinical trials to actually prove that these agents benefited patients. And that led to a number of companies being established in the late and mid-70s. The first biotech company established was Genentech. It was established by Bob Swanson and Herb Boyer in San Francisco. The third biotech company established was Biogen in 1978. And this is a picture of the Scientific Advisory Board of Biogen meeting in 1978. And the co-founders of that company were a number of scientists, 8 of us, myself leaning against the wall here. You will notice that I have a darker beard there and some hair. And Wally Gilbert there from Harvard, a professor who ultimately got the Nobel Prize for determining the sequence of DNA. Charles Weissman out of Zurich, a very distinguished molecular biologist. Bernard Mach from Switzerland. Hans Schaller, Ken Murray over here, a very international team. The company was originally established in Geneva, Switzerland in 1978 and moved to Cambridge, USA in 1980 or thereabout. And the motivation, again, for starting this company was to put together an organization that could both translate this science into something useful, but as well be able to train professionals in this new technology of recombinant DNA. And just to give you an impression of where that science has led over the intervening years, I'll use this example of the environment of MIT. You will see here the River Charles in Cambridge and Boston on one side, Cambridge on the other, and this red set of buildings to the north is MIT. And what you'll see around MIT in all of these bluish structures and all the names around it are biotechnology companies. There are at least 50 biotechnology and pharmaceutical companies located on the edge of MIT in Kendall Square. These companies range from companies of 50 and 100 people down to 2 or 3, but many of them are very large pharmaceutical companies that have moved into this area over the last decade. For example, there's Novartis, International Institute for Biomedical Research, the largest Novartis lab outside of even in Switzerland, I believe. Pfizer is located on the edge of MIT. Sanofi is located on the edge of MIT. And I will briefly talk about developments from the other 3 HOPs. This is the Broad Institute on the edge of MIT, Alnylam, which I mentioned before, and Biogen. Now, why are these companies located on the edge of MIT now? They're research divisions, mostly. And the reason they're there is this science is advancing so rapidly that actually to catch the cutting edge of the science, to be abreast of where the movement is in benefiting patients with this science, they need to be close to the academic community and engaged in that community in research. Let's talk about the Broad Institute. The Broad Institute is the institute in Cambridge across the street from MIT. It is an associated institute with MIT and Harvard. It's run by Eric Lander, and it is the major contributor of the human genome sequence for the completion of the genome sequence in 2003. This development of sequencing the human genome as a project started in 1990. It had cost $3 billion to get the completion of the first sequence in 2003. Yet today, you could probably buy your DNA sequence to be determined completely, even better than it was in 2003, for probably $500 to $1,000. The advancement of the sequencing of all types of nucleic acid information at the RNA, at the DNA, and in each an individual cell, which we can do now, sequence the content of each cell, has defined cell and molecular biology in new ways and given us fundamental insights into disease processes that are changing how we think about diseases and how we will actually begin to develop new therapeutic agents for it. But I want to take one simple example to illustrate a new development in agents that treat disease using DNA sequence, and that is human genetics. This slide was put together in 2015. We knew in 2015 there were 3,000 human genes, which, when they had mutations in them, we knew they caused specific diseases. They are called Mendelian diseases because they're segregating as individual genes in the human population. That number is probably close to 4,000 or 5,000 now. We only have 23,000 genes. So it's about a quarter of all of our genes now are marked by, if you have a mutation in this gene, this is the phenotype you will see. These are the medical problems you will or will not see. At this stage in 2015, it was considered that 8% of all live births were individuals who had a genetic disorder. Most of them did not know they had a genetic disorder. It's estimated that each child with a genetic disorder costs society about $5 million. Now that's a complicated number because it has to involve the care of the individual. It has to also involve the loss of wages of a normal, competent individual. And so that number is larger than you'd expend on medical care, but it is an estimate of what it costs society for these individuals. But the bottom line there is the interesting thing. Only about 11% of all children with genetic disorders were diagnosed with those as children. As adult, when the genetic defect is more apparent because of changes over time, 34% of adults who have genetic defects were sequenced and identified. But what this means is that with the advancement of human genetics and DNA sequencing, that diagnostic rate is going to move from 11 to 50, maybe higher, and the adult rate is going to double at least. So we are looking at a future in which genetic diseases are going to be a very prominent issue in how to maintain human health. That brings me to Emma Walks. Emma is about a 5 to 6 year old young child. She has spinal muscular atrophy, and before she was treated with the agents I will talk about, Emma couldn't walk, and Emma's life expectancy was very short. What's spinal muscular atrophy, which is a pretty common genetic disease of children? Spinal muscular atrophy is caused by a loss of function, a mutation or deletion in the SMN1 gene. It is a gene required for survival of the motor neurons in the body. And if SMN1 gene is defect, patients lose motor control, and that causes a number of problems, including inability to breathe. Now, in many patients, as you will see here to the right, there are other genes related to SMN1, in fact, very related, SMN2, in some cases SMN3 and others, that have the same DNA content, essentially, of SMN1, but due to a single nucleotide change, nucleotide change, which if I can get this pointer over here, has a splicing defect, that instead of splicing 6, 7, and 8, as you see here for SMN1, you splice 6 to 8, skipping 7, leaving out that exon due to that nucleotide change, and that gives a protein that's degraded rapidly. So if you're missing SMN1, and you're only making 10% of SMN2 as a functional protein, you have spinal muscular atrophy. Now, the treatment for this is to take an oligonucleotide that's about 20 nucleotides long, and as shown here as an orange bar, you can see it pairs with a DNA sequence within the intron of the SMN2 gene. And by blocking that sequence, switches the splicing of SMN2 so that instead of skipping that exon 7 in the previous slide, which would be the splicing pattern 2 shown in this slide, it goes through the splicing pattern 1, incorporating that exon, making a functional protein, and allowing children to walk and to actually have a pretty active life. Now, this oligonucleotide, which was developed by Ionis, as shown here, has to be injected into the spinal column every 3 weeks. But it allows these patients to now function in a reasonably normal life. That work was done by Adrian Craner, who's down at Cold Spring Harbor. He collaborated with Ionis, which is a biotech company developed to use antisense technology in 1989. And it was licensed to Biogen in 2012 and developed as a clinical product with the FDA approval 2 years ago, and it's called Spinraza. But what it says to us is that antisense oligonucleotides, when introduced into the spinal column, has exposure to a large fraction of the central nervous system. And we can think about treating diseases in the central nervous system with antisense or oligonucleotide pharmacology. Now, that integrates with another development in science, and this is the discovery of RNA interference, which happened in 1998, as was mentioned by Andy Farr and Craig Mello. Andy is the person to the left, and Craig in the middle. Andy is a professor at Stanford. He did this work when he was at the Carnegie Institute in Washington, D.C., and he and Craig published the paper in 1998 and received a Nobel Prize in 2006. And what they discovered was that double-strand RNA, most of the RNA we have in our cells, almost all of it, is a single strand made from one strand of the DNA. But if by chance there's 2 strands of that RNA, the cell will break down that double-strand RNA and use bits of it to actually silence the gene that the double-strand RNA was made from. Double-strand RNA is an alarm molecule in the cell. We didn't know that till 98. We didn't know that our cells had this whole pathway until 1998 due to the insights of Farr and Mello. At MIT, I and a number of colleagues, Dave Bartel, Phil Zamore, and Tom Tushel, worked out the chemistry of this small RNA and set out to actually use the small RNA as a therapeutic agent, as a double-strand RNA, to treat diseases. So what we knew at the time in 2000, 2001, is that the double-strand RNA would enter the cell. If it entered the cell, it would be unwound. One strand would be loaded into a professional machine in each of our cells. The primary molecule, protein, is called argonaute that holds it. And that then would search the cell for complementary RNA sequences from a messenger RNA, that intermediate between DNA and protein. And if it found a complementary sequence, it would then cut that sequence, destroy the messenger RNA, and silence the gene. So what this suggested is if we could actually make this small RNA process work, we could take the sequence of the gene, design the small RNA, introduce it into cells, and silence disease genes that was causing problems of patients. So we set out by establishing Alnylam in 2002. And I'll show you the progress we've made over the past 16 years of science trying to advance this technology to treating diseases. And I'm going to talk about the treatment of this disease, transthyretin, which is a protein folding disease. Transthyretin is made from a gene in your liver. It circulates through your blood system carrying vitamin A. When it has a mutation, it can misfold, and if it misfolds, it accumulates in your nerves and neurons, as shown by the staining there to the right, and that causes loss of motor neurons and loss of cardiomyocytes, and therefore loss of heart function and nerve function. And from patients who have this genetic defect, they begin to show symptomology at about 40 years of age, and typically the disease process is fatal within 5 to 10 years. So we could easily make these bits of RNA to the gene that's expressed in the liver, but we had to deliver the RNA into the hepatocyte, the cell in your liver that makes transthyretin. And to do that, we used a small lipid particle, a lipid nanoparticle, which is a small particle about the size of a vase, which we made by coating the RNA, as shown in that little sphere to the left, with a lipid coat, so that the lipid coat could be fused to the lipid surface of the cell, and by that means release this RNA into the cytoplasm of the cell. This had really never been done in a patient, and we were then exploring how this could happen. And what we recognized is that once the particle enters the vicinity of the hepatocyte, as you see it in that surface called a cell, surface of a cell, hepatocyte, it has to be invaginated into an endosome. It's a little vesicle within the cell, and those membranes have to fuse to release the RNA into the cell. So how could you make these membranes fuse? And what we found was that membrane fusion could be enhanced if we could change the topology of the lipids that were part of that particle. And what you see to the left is if you have two lipid with charges at their surface, and they're vertically and unpaired to each other, and that would happen at certain pHs, they form a bilayer structure. But if you then change the pK so they're attracted to each other, they will change their topology, and the lipids will flip to this hexagonal array structure. You can test that in your laboratory as a test tube. And that change in lipid structure promoted the release of this RNA into the cell. One of the hints that this was an important issue was when Alnylam made lipid particles with different pKs on their acid and base, positive and negative charges, and tested the efficiency of silencing a gene in the liver of a mouse, and that's what's shown on this slide, and this is the efficiency of silencing along the vertical axis, the pK of the interaction of those charges on the horizontal axis. And what you clearly see is that at a pK of 6.4, slightly acidic, 6.4, the efficiency increased of silencing the gene in the liver by 2 orders of magnitude. 2 orders of magnitude is an enormous amount, because what it meant is that we could change the dosing of the siRNA, the small RNA contained in that lipid particle, by 100-fold and get the same amount of silencing in the liver. And that reduced side effects by 100-fold, the amount of material you had to introduce into the patient by 100-fold, and therefore made it possible to use these types of technologies, these small RNAs, as a therapeutic modality. This is the results from a phase 3, which is after you've shown efficacy at toxicity at phase 1, efficacy at phase 2. Phase 3 is for approval. And what you see here is a diagram over about 2 years of patient treatment. The red line is a placebo-treated patient, same patient population, but not receiving these lipid particles with a small RNA to transthyretin. And the blue line is the treated population. And you're looking at the serum level of this protein, TTR, that accumulates in the tissue and causes the disease pathology. And what you can see is there's an 80% reduction in the level of the circulating protein over a 2-year period in treatment of these patients. So you infuse these particles into the IV into patients every 3 weeks or every month, and you can see sustained reduction in these patients of that circulating protein that causes the disease pathology, in this case, neuropathy. And what you see when you follow the patients is shown here on the left. This is the results of the phase 3. The drug is called patisiran. It's a lipid particle delivered small RNA. And what you can see over 18 months of treatment is if the patient is receiving the placebo, that's the red line, and they are progressing to lose their ability to walk and be active, and that's what this MS+5 indicates. But if they're receiving the drug, they're actually improving. At least half of them are gaining more activity levels. And if you look at the Norfolk quality of life indication, you see again the patients receiving placebo are progressing, whereas those that receive in a double-blind trial the drug are half of them are getting better, more active in life, better able to take care of themselves. So this treatment was approved this year by the FDA in the US and in the EU for treatment of TTR for neuropathy. And it's the first small siRNA drug approved in the world. So it took us 16 years. In fact, from the discovery, it took us 20 years to take this new insight of how small RNAs can silence genes in cells to a therapeutic indication of benefit to patients. But we expect this to be increasingly common because there's another method of introducing these small RNAs, and that is, as shown here at the bottom of this slide, where you see the small RNA as the 2 parallel lines, as 21 nucleotides long, and we have attached it is attached to a sugar here. This sugar is GalNAc, it's a 3-headed sugar, and it turns out that on the surface of your hepatocytes, there is a receptor for that sugar that gets internalized, and that's shown in these bind to the surface, get encapsulated into an endosome, and then get released inefficiently and get released into the cytoplasm. This particular treatment you can administer sub-Q. And the striking thing about it is that, as shown here for another clinical trial, not a phase 3, but a phase 2 clinical trial, you have PCSK9, which is a controller of cholesterol in your blood, and with one dose of injection of that sub-Q at 300 milligrams per patient, you see prolonged over 5 months silencing of that gene. It will return to normal levels after about 6 or 7 months, but a prolonged silencing of a specific gene in the liver with one treatment every 3 to 4 months. So I think siRNA is going to be an important modality for treatment. I think antisense oligonucleotides are an important modality for treatment, and a whole new translation of science based on structure of genes, splicing patterns, insights into RNA interference is offering us a whole new modality for treatment of critical human diseases. I want to end with a startling observation and a new concept in science that I think will be very important over the next decade or so as we integrate our knowledge of genes and gene regulation and disease processes through the lens of a new property that we just recognize exists in cells. What is shown to the right of this slide is a cartoon of a cell. We have been looking at cells with light microscopy for 100 years. And we have noticed in the cells dark, dense areas called the nucleolus, the Cajal body, after the great Spanish scientist, the P bodies in the cytoplasm, and many other bodies that are listed there. These are concentrated ensembles of proteins and RNA that are concentrated in certain regions of the cell, but they do not have a membrane about them. They're concentrated by not being contained within a membrane. They're concentrated by other forces. And it was Tony Heinemann, who's in Dresden, Germany, and Cliff Brandywine, who was a postdoc at his laboratory, now at Princeton, who looked at these bodies in a simple worm called C. elegans and recognized that these bodies had the properties of fluids, that when you watched them under the microscope, they fused and fissioned. If you put them in shear forces, where you move liquid across the cell, they deformed. So they were not solid bodies, they were liquid-liquid type bodies. And they called them liquid-liquid type phase transition bodies. Well, that suggested a whole new physics of how these bodies formed and could possibly suggest a new physics for how many of the processes occur in cells. So this happened in 2008, 2009, and over the last several years, and in fact this year, it is becoming clear that these types of processes are involved in a wide range of critical cellular processes in cells. I'm going to talk a little bit about one specific process in cells that is central to the activity of cells in the last few moments. But just to give you a sort of schematic view of how these membraneless particles form, I want to show this experiment, which is from Michael Rosen and Roy Parker. And what they did was to take an RNA binding protein shown at the top of the slide to the left, which is RRM. That's an RNA recognition motif that binds to certain sequences on RNA, and there's 2 of them, and then an intrinsic disordered domain. And we've puzzled for decades when we got the sequence of many of the cellular genes that are involved in gene regulation and RNA metabolism as to why they had these disordered domains, these unstructured, repetitive domains on them. They showed that they mixed this with RNA, And because those intrinsic domains could interact with each other, and these RNA binding motifs could bind to the RNA, that at a certain concentration of that RNA and protein, you got formation of one of these liquid-liquid type phases. And they concentrated the protein and RNA about 100-fold in this membraneless body as compared to the solution around them. And you'll recall from your basic chemistry that if you have something concentrated in a reaction, you can get a more rapid reaction. You can get more facilitated interactions. So working out the chemistry of this, what we showed is shown here. We took a simple model in which we had chains, and this is a collaboration with a physicist at MIT, Arup Chakrabarty, and his graduate student. And we modeled it as having sites in which modifications occurred. So we wanted to be able to theoretically vary the valency, which is this F parameter, and we modeled that by having sites of modification. And every site of modification can cross-link to another site of modification, and that's a binding coefficient in this model. And that's a K on, K off, as you see there. And the K on, K off is the binding coefficient shown at the bottom. And now you can ask, under what conditions does these molecules go from a random solution to a gel-type liquid-liquid phase? And if you solve that problem, what you note is that a critical concentration over the number of chains in, how big this ensemble is, at what concentration it exists in the whole solution, varies as 1 over the binding coefficient, but varies 1 over the square of the valencies. These interactions are driven by valency. How many cross-links, how many links different macromolecules can make with each other. Once you get to a certain level of valency and affinities, you form these gels. And those are condensates, we'll call them condensates. And those are the functional units in these reactions. So I want to illustrate one process in the cell that is at the heart of how these cells work. And this is then control of the genes that are expressed in different cell types. You will recall from your basic biology that once you get a fertilized egg as a zygote, it undergoes embryogenesis through the blast stage and then through a fetal stage to give rise to all the cell types in the body. And each of those cell types, as illustrated to the right, has master transcription factors that are unique to each of those cell types that derive the expression of critical genes in that cell to distinguish a skin cell from a blood cell, from a muscle cell, from a neuron. And those have been called master transcription factors by my colleague at MIT, Rick Young. And he recognized that adjacent to those master transcription factors, there are typical DNA sequences, regulatory sequences, critical sequences, called superenhancers, who I will describe in a moment. The critical point here is that the master transcription factors bind to those superenhancers and talk to each other to maintain the homeostatic identity of the cell. How do we know they talk to each other, and how do we know there is a super enhancer? And that's illustrated here to the right and left. What we do in this experiment is take cells, fix them so the proteins don't mix, and then isolate DNA and protein, break up the DNA into small pieces, immunoprecipitate out the protein, and ask where's the protein bound by DNA sequencing in the cell. And what you see here to the left is a typical enhancer for a gene that's expressed at low levels in the cell. And its promoter, at the bottom of the slide, you see the gene. A typical enhancer has these OX/SOX/NAN transcription factors bound. It has polymerase bound. It has two other proteins I'll point out BDR4, part of the process of formation of proteins at this enhancer and promoter, and as well mediators shown here at the top as MED1 and MED12. And you see on the left that there is one of those enhancers for this gene, but on the right, for a gene expressed at high levels, there are 6 or 7 of those enhancers. And that's called a super enhancer cluster. And this gene is expressed at high levels because of that super enhancer. Let me quickly show you some properties of the super enhancer that distinguishes it further from this single enhancer. First, if you ask, do the pieces of the super enhancer where the DNA is bound, do they all coalesce in one concentrated region? And we can do that by basically asking, do DNA sequences from one place appear in the cell close to DNA sequences from another? And you can see by all those lines above, that yes, that is the case. They're all talking to each other or in a close association in this super enhancer with the promoter. And then we can ask another question. If you start and change the level of the activators, and you see those are the blue dots in this slide, for a typical enhancer, as you increase the level of an activator, the promoter and gene shown to the right increases linearly. But if you do the same for a super enhancer gene, this is a different gene driven by a super enhancer, what you see is a sigmoidal. You see more activity than you would expect from a summative effect. You can inhibit super enhancers and show this as well. This drug, JQ1, which is shown to the left, and we're measuring the binding and activity from a super enhancer on the left side, and that from a typical enhancer on the right. And I'll bring your attention down to 500 nanomolar, and what you see is there's a 30-fold decrease in activity with the super enhancer, and only a 3-fold decrease with a normal enhancer. So it's cooperative and inhibiting, showing increased sensitivity to these types of drugs. So we know super enhancers are large clusters of factors. They have properties that look like these liquid-liquid clusters. They are highly sensitive to deletion of subunits, and they're inhibited more intensely by drugs than normal enhancers. I won't talk about the bursting, but I'll show you in the last slide, which is a few moments from now, how we see that. So we know that these super enhancer factors are found in the cell in discrete bodies. I will show you that in the next slide. And I also show you that the super enhancers co-localize with the genes that are known to be associated with super enhancers by activity. And what we're asking here is, what are the properties of the super enhancer that looks as if they're these liquid-liquid phase condensates? Because if they have those properties, I understand something about their physics, I understand something about how to drug them, and I can gain insight from that in terms of understanding how genes are regulated in the cell. And what this shows you for 3 different genes, all expressed at very high levels in this embryonic stem cell, is that if you look at the cluster for BR4, that's green here to the left, in 3 different genes, you see the cluster marked by a square. If we identify RNA from genes known to have super enhancers, they co-localize with that cluster, showing you that a cluster of 10s and 50s of these factors in one place, a condensate, is associated with a gene that has the properties of super enhancers. So that's one property of super enhancers. Another property of super enhancers is that they're dynamic. Factors exchange in and out of these condensates very rapidly. If they're a liquid-liquid phase transition condensate, factors exchange in and out of them very quickly. And you can actually ask that question by the following experiment. You take components of that that condensate, in this case, the little blue dots. You label them with a protein, green fluorescent protein, that you can image, and you can find them in clusters, and then you use to photo bleach those clusters so they don't fluoresce anymore, and you ask how fast they can recover fluorescence. If they're dynamic, they will recover fluorescence quickly. It's exchanging factors with the factors in the solution around them. A very dynamic, moving, pliable, liquid-liquid type condition. So here you see on the left the BDR4 GFP at one of these super enhancers. The arrow points to it. You point a laser and you bleach it, and you ask how fast does it recover. And what you see here to the right is that it recovers in about 10 seconds. So that whole condensate is dynamic in the sense that its factors are exchanging with other factors around it in the cell in a time scale of 10 seconds. 10 seconds, a very dynamic property. Now, I told you transcription happened in bursts. This is where technology is beautiful. Technology is changing everything we do. And here, the technology is, can I watch in a live cell the super enhancer kissing the gene? Can I see it kiss? And this is the work of a colleague here at MIT, Ibrahim Sissi. And what he's done, as you see here in the diagram up there, is he's labeled a component of the super enhancer called a mediator with the green fluorescence. He's labeled the gene with the orange or bluish fluorescence. And what he's imaging here in the cell, in a live cell, where he can watch it, is the separation of the two. So what you notice here in this diagram to the left at 1300 minutes is that the green and the purple are separated. You watch the cell for another 5 minutes or 7 minutes at about 20 minutes, and you see they move to kissing. That's 18, 5 minutes later. At 37, they're still kissing. Long kiss. And at 43, they've separated again. And this super enhancer with liquid-liquid phase transition properties is associating with the gene, and during that association, it is bursting in gene transcription, giving you regulation of the expression of that gene. So we know super enhancers have large ensemble of factors and enhancer RNAs, large valencies. They contain factors like mediator and BRD4 and polymerase that have these intrinsic disorder domains. They are critical components in forming these condensates, and we are confident and increasingly confident that this whole property of condensate formation is at the key of how these critical genes that control cell type identity, the differences between blood cells and brain cells and skin cells, is in this property of forming liquid-liquid phase transition clusters. In the last year, we now have publications that say synapses, the connection between your nerve cells, the reason you can think, the reason you can listen to me, those synapses have proteins that have liquid-liquid phase transition. We don't know it's controlling the synapse, but I'll bet you it's controlling the synapse. The spliceosome process that removes introns from genes have liquid-liquid phase properties. Stress granules, which control how cells respond to stress. Cells respond to stress all the time, have liquid-liquid phase properties. So we think there's a whole new way of looking at our biology. Tony Hyman and Cliff Brandewine introduced it. We've shown it for a transcriptional process. And I think that'll lead us to new insights into how to develop therapeutic agents that target this valency, this critical issue of interactions, and therefore could be used to treat and understand disease. So I've taken you through an adventure of about 50 years, of the discovery of the structure of DNA, to molecular biology, to cellular processes, to now phase transitions, and illustrated in all of those cases how fundamental science, advancing our knowledge, has changed and empowered us to understand disease processes and treat disease processes. I thank my colleagues in my research group for their help, particularly the collaboration of my colleague Rick Young at MIT and Arup Chakrabarty at MIT, which makes life incredibly enjoyable to have scientific colleagues who can get together and create new ideas. And I particularly thank the laboratory of Rick Young for the super enhancer liquid-liquid phase transition data. And I thank you for your attention and for this invitation.
Thank you, Professor Sharp, for this clarifying story full of innovations and promising perspectives. So now we are concluding this act. We are going to receive the Chamber Chorus of the University of Salamanca. So I don't know, the volunteers, do you have to take this off, please? Thank you for your wait. We are now concluding this act, please. Can you hear me? No. No? No. Can you hear me now? Yes? We are concluding now this act, so I wish you a profitable Congress full of scientific exchanges and personal experience. And now we are receiving the Chamber Chorus of the University of Salamanca. After the chorus, we are going to move to Colegio Fonseca to enjoy the welcome cocktail. So first, receive the Chamber Chorus of the University of Salamanca.
Muy pocas cosas, no se preocupen, de lo que vamos a hacer hoy. Nuestra idea era traerles a la Salamanca del 1500 aproximadamente, a la época de Juan de la Encina, Él se fue de Salamanca un poco antes, se fue en 1598 porque opositó a una plaza de cantor en la catedral y no la obtuvo. Entonces, un poco dolido, se fue de Salamanca sacudiéndose el polvo de las zapatillas y se trasladó a Roma. ¿Y por qué? ¿Por qué cantamos esto? Porque el códice más importante posiblemente de música profana, hispana e ibérica, diría yo, del siglo XV y XVI, de finales del XV y principios del siglo XVI, es el Cancionero de Palacio, que en él están la mayor parte de las obras que vamos a cantar hoy. Y una de las hipótesis es que el Cancionero de Palacio está vinculado al Colegio de Anaya, al Colegio de San Bartolomé, uno de los colegios mayores de Salamanca, y por tanto su repertorio sería un posible repertorio estudiantil. He de decirles que en realidad yo estoy llevándome el agua a mi molino, esto no está demostrado del todo. Hay otra hipótesis que lo vincula a la corte aragonesa de Fernando el Católico, otra que lo vincula a Medina de Río Seco, por cierto, a un almirante, al almirante de Castilla, pero si no es verdad, nos permiten la pequeña trampa. Y hemos dividido esto en pequeños bloques, donde Juan de la Encina, insisto, del que celebramos ahora en los 550 años del nacimiento, está muy presente. El primero ya han oído, es un consejo, todo pasa, pero lo que se queda es la fama y la gloria. Yo creo que no es mal comienzo para un congreso como el que van a celebrar. Y el siguiente es un llanto por la muerte del príncipe Juan. El príncipe Juan murió en Salamanca en octubre de 1497, creo recordar. Su pendón preside nuestro paraninfo y es para mí es uno de los llantos más hermosos. Verán que tienen las letras por ahí, porque creo que merece la pena las letras de Juan de la Encina. Él era, aparte de un excelente músico, un singular compositor. Un poeta, perdón. Muchas gracias. Y en el cancionero de Palacio, como en casi todos los cancioneros, el tema fundamental, el tema principal es el amor, y al amor van dedicados los siguientes bloques del programa, pero distintos tipos de amor. Y al final alguna cosa que se parece algo al amor, pero no tiene mucho que ver. El primer bloque es las tristezas del amor. Y vamos a empezar con una obra de Francisco de la Torre, muy breve. Esta obra de Francisco de la Torre, que es un autor que también está en el cancionero de Palacio, sin embargo pertenece al cancionero de la Colombina, que es un poco anterior, unos 15 o 20 años anterior. Y habla bueno, que la pena de amor es mejor que el no sentir nada, ¿no? Más vale sentir el dolor del amor que no tener amor en absoluto. La siguiente es una de las obras más conocidas de Juan de la Encina. Es una canción pastoril y cuenta la historia de un pastor que va el jueves al mercado y se enamora. Requiebra a la doncella, pero ella no le hace caso. Dice literalmente, aballó la pata. Y a partir de ahí él lamenta su fracaso y vuelve a hablar de las tristezas del amor. Y la tercera pieza, Más vale trocar, si les recito la primera estrofa, pues dice exactamente, más vale trocar placer por dolores que estar sin amores.
Porque callas tú
el siguiente. El siguiente tópico va a ser el amor cortesano, con dos piececitas que vamos a cantar ligadas ambas, y que además ambas están escritas en una mezcla de lenguas, en una mezcla de castellano, de francés e incluso de italiano. Y en ambas también el ambiente es un ambiente muy bucólico. En el jardín es bastante frecuente los encuentros del amado y la amada en el jardín. Esa es la primera pieza. Y en la segunda, además, la amada le pide a un ruiseñor que le lleve un mensaje a su amado. Y el mensaje es triste, que es que ya está casada. Y el último tópico del amor propiamente dicho, luego vendrán otras cosas, es el amor guerrero, el amor que es capaz de romper con cualquier cosa, de asaltar las fortalezas más fuertes. Y Encina escribe un romance maravilloso que se titula Mi libertad en sosiego y que acaba que está lleno de esos símiles. Y acaba ese romance con una deshecha, con un villancico que se titula Si amor pone las escalas. Si amor pone las escalas al muro de la ocasión, no hay ninguna defensión. Tengo tantas letras en la cabeza en este momento, disculpen. Bien, sin duda, El amor formaba parte, seguro, de la vida estudiantil, como intuyo que ahora también. Y lo que vamos a cantar ahora creo que también formaba parte de la vida estudiantil entonces y creo que ahora también. Hay varias teorías sobre esta canción cuya letra es ininteligible, como van a poder ver, pero hay algunas alusiones muy claras a la bota que se mea, dice exactamente. Es una canción que sin duda es una canción báquica, pero una de las hipótesis es que es una canción de trabajo marinero, que también se cantaría en las tabernas. Es la tricotea de autor anónimo y que está en el cancionero de Palacio. Salamanca, por su universidad, fue un centro religioso importantísimo. La mayor parte de las comunidades religiosas tenían un convento en Salamanca y muchos de los profesores de la universidad y de los estudiantes de la universidad eran religiosos. Entonces, hacer una referencia a los frailes es inevitable y a las dueñas. Pero las referencias que hay en los cancioneros a los frailes y a las dueñas son... La primera, Pinguele Respinguete, que está también en el cancionero de la Colombina, habla de una señora que su marido se fue con el arzobispo a servirle y le dejó un hijo y cuando volvió encontró que tenía 5. Pero de esos 5, 2 eran del convento del Carmen y 2 del convento de San Francisco. Esa es la primera historia. Y la segunda historia es de un fraile flaco que, bueno, la van a oír.
medias y pañales, vendió, empeñó las decretales, y en vano con todos sus males, no tiene juicio divino. La moza que ve livianilla, no desarmó en fraile de Sevilla, y hasta tomar la lucilla, ya más no la pierde del tino. De aquel fraile flaco y cetrino, guardaos dueñas del que es un maligno.
Era una buena pieza. Y para acabar, dos canciones de algo tan viejo como el mundo, que son Los Cornudos. La primera de ellas, además, es de Pedro Fernández, que curiosamente se llama como uno de los tenores del coro que trabaja en la Facultad de Farmacia. Pedro Fernández efectivamente fue un músico activo en Salamanca a finales del siglo XV y principios del siglo XVI. ¿Tienes que ver con él? ¿No? Y da unos cuantos consejos para no ser cornudo. Y la otra cuenta una historia, la segunda obra es de Juan de la Encina, con la que acabaremos, y cuenta una historia real. Juan de la Encina, como les comenté al principio, se fue a Roma. Y en Roma uno de los músicos que era uno de sus rivales principales era Bartolomeo Trombonchino. Y Trombonchino, en junio de 1499, cuando llega a su casa, se encuentra con que su mujer estaba con otra persona, con otro hombre. Y, encolerizado, la mata. Esto es cierto, se cuenta hay constancia de que fue así. No es el único músico que mata a su mujer, por desgracia ha habido alguno más. Y entonces Juan de la Encina cuenta la historia, porque esas cosas, siendo su enemigo, se tienen que saber. Y está escrita, falta la parte, está escrito en un italiano soldadesco, como verán.
Guarda, no lo seas tú, guarda, no lo
Excelente repertorio y magnífico concierto. So we are moving to Colegio Fonseca to enjoy the welcome cocktail. And just remember that tomorrow we start at 9 o'clock at the Faculty of Pharmacy. Thank you, everyone.